Первый доклад будет посвящен применению позитронно-эмиссионной томографии в иммуноонкологии. Это очень интересное и перспективное направление, которое сейчас бурно развивается и связано непосредственно с технологиями тераностики. И действительно, тераностика, сейчас много о ней говорят, очень будем надеяться также, что она придет к нам в Россию тоже. Для этого есть все предпосылки. Второй доклад будет посвящен технологиям позитронной эмиссионной томографии, технологиям, это понимаю, Healthcare. Тогда я позволю представить первого докладчика, Бен Ньютон, продакт-менеджер Healthcare. Доклад называется «Иммуноонкология. Почему без ПЭТ нельзя?».
So thank you very much for the kind introduction. My name is Ben Newton and I lead the oncology business at GE Healthcare. I would like to talk to you about some of the developments that we have at GE Healthcare to support oncology diagnosis and prognosis using molecular imaging tools, and also some of the ways that we are developing solutions for our customers. It used to be that our focus was simply on equipment and developing high quality equipment and high quality products for use in those equipment, contrast agents, molecular imaging tracers. But now the changing world in oncology, particularly the complexity, the workload, has led us to focus on not just developing the equipment and tools for diagnosis, but also solutions that will help implement those tools and equipment and help improve the workflow of patients through the diagnostic care pathway of oncology. In addition, because of the increasing number of patients and the increasing complexity of disease, we're developing IT platforms as well. IT platforms that will support the integration of data, molecular imaging data, pathology data, genomics data, and so on. And these developments, these solutions and technologies, we intend to position to support the delivery of care and improved outcomes. Because it's not just about selling equipment, it's about improving the lives of people, improving the outcomes. And our aim in the future is to deliver precision health, supporting you and enabling you as physicians to deliver precision health for your patients. In addition to this rise in acceleration in cancer, particularly lung cancer, prostate and breast of course, is the challenge of innovation. We're in a world of constant innovation, constant development of genomic tools, analytics tools, radiomics tools that are affecting all areas of the care pathway. Gone are the days where simply taking an image in response to a referral from an oncologist was enough, and then the prescription of chemotherapy was the only option in cancer. Now, of course, there are immunotherapies, cell therapies, radiation therapies. Around 60% of patients nowadays get radiation therapy. And now we see proton beam therapy, radiotherapy, treatment of solid tumours using targeted radiotherapy and theranostics is all taking part in this revolution of cancer care. And with this revolution is coming an increased complexity of data. And the management of this data and making sure that this data is informing on the right kind of choice of treatment is part of what we need to help and support and enable for our customers. So the opportunities that we see for supporting customers really are in three areas. We can support customers through the design of innovative care pathways, the ability to re-engineer and support service delivery, new business models to deliver care in new hospitals, in new clinics, in a rapid diagnostic context. You know, if you can speed up the diagnosis after screening and you bring the treatment closer to the diagnosis, then you can change outcomes for the better. It's well established that if you move from diagnosis from stage 3 or 4 in cancer to stage 2 or 3 and treat appropriately, the outcomes are certainly improved. So there are 3 areas that I wanted to focus on, a little bit around our molecular imaging portfolio, what we have currently, and how we're developing that in immunotherapies to support the selection of patients, the stratification of patients, and the monitoring of patients with immunotherapies using molecular imaging tools. A little bit about how we are changing the care pathway and speeding up diagnosis and bring therapies. And then finally a little bit around decision support with the I2 tools and platforms that we're developing. So many of you will be familiar with our workhorse for the production of radiopharmaceuticals, the FAST Lab. We have developed over recent years 2 products, Visimil, an amyloid PET tracer for the diagnosis of Alzheimer's disease, and more recently we're in the process of seeking approval for flurpiridaz, which is a cardiac PET tracer for the detection of myocardial perfusion. Both these agents are not currently registered in Russia, but we're seeking partners for the development and approval of these agents, and also partners in the exploration of the clinical utility for these agents in Russia. I should point out that flurpiridaz is an extremely advanced agent, very similar in profile to the likes of O15 water in being able to linearly and directly correlating with perfusion of the myocardium, even under high stress conditions. So these novel agents are complementing other agents that we have in the portfolio that are available on the cassette, including agents like FDG PET, increasingly gallium PSMA, fluorine DOPA, as well as other neurological agents like G180. I should say that what's driving this transformation in the portfolio is not just the development of new tracers and targeting agents, but also the availability and abundant availability of gallium. Gallium, currently produced on generators, can be produced really cost effectively, but in low volumes. The transformation of solid target technology of the cyclotron is allowing us to lower the cost of production of those and increase the concentration and volume of production of gallium-2. So the ability to bring gallium at a low cost, high concentration, in conjunction with this tracer technology is revolutionizing not only the variety of tracers that we can produce, but also the speed with which we can develop these tracers too. So coming back to the portfolio, I've mentioned a little bit about cardiology and the PET tracers for myocardial perfusion. We'll go back. We have PSMA and HER2 on the oncology front. I want to talk about these 3 tracers, the immuno-oncology tracers for the selection of patients who will undergo PD-1 or checkpoint inhibitor therapy, or CDH tracers that will allow the assessment of immunocompetence and also the monitoring of therapy. There's been a transformation in treatment of cancer in recent years with the advent of immunotherapies. Products like Opdivo, Keytruda from BMS and Merck have transformed outcomes, and as you see here, compared to chemotherapy in advanced non-small cell lung carcinoma, the survival rate in anti-PD-1 therapy is transformational, and led to the award of Nobel Prize in Medicine in 2018 for Jim Allison and Professor Honda here. But the problem with this therapy, although it is transformational, can be that only a small minority of patients can be responsive. Around 20 to 30% of lung cancer patients are responsive. The responses are durable in those cases, but even in those cases there are some strong side effects. So actually selecting patients that are going to be responsive, maybe deselecting patients that are not going to be responsive, and if patients are responsive and side effects could emerge, and these agents do develop side effects, making sure that the therapeutic index is extremely important. So what we are embarking on with our program in immuno-oncology is not only developing molecular tools to stratify patients such as PD-1 tracers and CD8 tracers. We're also looking at artificial intelligence-based methodologies for the prediction of responses, prediction of side effects too. And that's important because one of the big challenges in detection of therapeutic efficacy using current approaches is the problem of pseudoprogression. Illustrated here, this metastatic melanoma solid tumour at baseline imaging seemed to be advancing in 3 months even after anti-PD-1 therapy. In fact, what happened 3 months beyond that was a resolution and a reduction in tumour size. And this pseudo-progression is manifest because the immune system has been activated. The checkpoint inhibitors are doing their job. They're developing a host response, inflammatory response to the tumour, and that is promoting tumour cell death and resolution, and what could be a complete response. But it takes 3 months, 3 to 6 months for such a response. Generating a marker such as a CD8 T-cell marker that can track the influx into the tumour within a matter of days could not only allow decision-making around the efficacy of the immunotherapy, but also allow us to get a very clear health economic proof that it's worth continuing that therapy in those patients. So this is the mainstay, and we are still developing radiomic tool and artificial intelligence-based methodologies to explore the texture changes in this kind of tumour to this kind of tumour, but complementary approaches around the use of CD8 detection and monitoring of therapy could give us a very rapid means to assess efficacy within a few days, and actually could also give us the tools to detect susceptibility or presence of immunocompetence in clinical trials as well. And this is the reason why immunocompetence is a requirement for response, and immunocompetence is driven by the presence of CD8 positive T cells. So having those in close proximity and being able to be activated is in large part a predeterminant of response to PD-1 therapy, and so detection of them is critical at the start and then of course through the therapeutic response. So the probes, as I briefly mentioned, that we're developing, and as yet these data are confidential, but we'd be very interested in partnerships in this space. We have infiltration markers, as I said, for CD8. We're also developing markers for T-cell activation, because in addition to the need for T-cells to come into the tumour to kill off the tumour, we need those cells to be activated as well. So having a marker of activated T-cells could give us an even more enhanced way of determining that there is a physiological response to the therapy. And finally, on the companion diagnostics space, developing tracers that could in situ and non-invasively assess the PD-1 or PD-L1 expression in the tumour, again with molecular imaging, could be a means to stratify for eligibility, not only for clinical trials with checkpoint markers, but also for clinical treatment in clinical practice. So this portfolio has been activated a few years ago, and we have developing data sets that we're interested in developing further clinically and also regionally too. Just briefly on the side effect side, as I mentioned, side effects such as pneumonitis, cardiotoxicity, even diabetes is resulting from checkpoint inhibition. So being able to predict which patients potentially could be susceptible to these side effects is extremely important in optimizing the therapeutic index. So in conjunction with a partner in the US at Vanderbilt University, we are retrospectively analyzing data, longitudinal data over a number of years from patients that have been treated with combinations of immunotherapy and chemotherapy, and we're exploring the relationships between the genomics and the treatment outcomes to assess which patients actually were more susceptible, depending on their genetic background, to different types of therapy. And these are the kind of use cases that we're exploring prediction of resistance to immunotherapy, prediction of response, obviously prediction of toxicity, as I mentioned, and eligibility of patients into immuno-oncology trials. So changing tact a little bit, these technologies and tools for imaging and stratification and monitoring of therapy are extremely important in the selection of drugs and the selection of right therapies for patients. But what about diagnosis? What are we doing about improving the speed of diagnosis? Well, one of the areas that we're focusing on is in breast cancer, where screening has been established for many years. And the other area is in lung cancer, where screening is perhaps not so well established and somewhat problematic, but the diagnosis using low-dose CT can be extremely important and powerful in identifying at-risk nodules and therefore leading to selection of therapy. So we're working with a number of approaches, one of which is in the case of breast cancer, with this one-stop clinic, which we established a few years ago in partnership with the Gustave Roussy Centre in Paris. And in the one-stop breast cancer clinic, we're able to compress the whole process of diagnosis to treatment planning in one day. Now, most of the world, and in most examples, screening which takes place in at-risk or susceptible patients is organised through screening programs, and the process and timeline from diagnosis to treatment planning can be as much as 6 weeks. At Gustave Roussy, we've brought that down into a single day for 75% of the patients, and that's leading to a complete transformation in the outcome of patients, because in that centre we can dial in the correct therapy on that same day and start treatment extremely quickly. So just to take you through some of the data, 75% of women coming into screening are diagnosed on the same day. We'd like it to be higher, but some patients require more investigation, they require MRI. And those MRI and other investigations beyond ultrasound and mammo are usually carried out on day 2 or day 3. So even with those more complicated advanced cases where they're diagnosed within a couple of days. The main advantage of this is not just the sensitivity and specificity of diagnosis, but actually the anxiety reduction and the improved experience for women. And the majority of women in these circumstances actually are negative. The majority of women having an abnormal screening profile are negative upon diagnosis, but it's an extremely anxious time. In addition to that, we've brought the time down using fine needle aspiration, cytology of the breast cancer lesion rather than core needle biopsy, processing with immunohistochemistry. And this leads to not only a time reduction, but also a cost reduction. We've since now templated this approach into Colombia. I'll show you the data, and now we're moving into the US to template that across 4,000 hospitals in the United States. And just to give you the status in Colombia, the previous time between suspicious lesion and diagnosis was 65 days. We're bringing that down and have now completed screening and diagnosis of 100,000 women in Colombia. And this is now becoming part of standard practice in Latin America. So moving quickly to the lung cancer situation, again, the challenge in lung cancer is not so much the fast diagnosis and time to treatment. The time that this is not easily visualized, but these are the improvements in survival over the last 20 years. And you have to believe me that this is the improvement of survival of prostate cancer increasing substantially and female breast cancer increasing substantially over the last 20 years or so. So these diseases, because of good screening practice and rapid diagnosis, is transforming those cancers into more chronic conditions. If you look at what's happening with liver, pancreas and lung down here, the survival rates have hardly changed over the last couple of decades. And that's because we're not screening very well, we're not diagnosing fast enough. So our rapid diagnostic centres that we're establishing in the UK is an attempt to replicate what we're doing in breast cancer. And here we are setting up rapid diagnostic centres for patients who are referred by their general practitioners following perhaps repeated cough, looking like some sort of viral cough, but they can be referred for sputum assessment and maybe even biomarker assessment as screening, and then referred for low-dose CT. And if you're unfamiliar with the data on the benefits of low-dose CT, I can certainly send you some of the more recent findings from the Nelson trial and other recommendations where lung cancer outcomes are improved substantially through low KVP CT screening. So great examples there of how not only we can practically bring the tools to select patients, to better treat patients more precisely, but also actively bringing those tools and products into better solutions to speed up diagnosis and target treatment. So I mentioned right at the start that it's a highly complicated process now in oncology. The number of genes associated with cancer is becoming bigger each day. The complexity and the burden of information that oncologists, particularly medical oncologists, have in making a diagnosis and tailoring the treatment is a huge challenge. And likewise, for radiologists, in MDT scenarios, radiologists have to attend multiple multidisciplinary team meetings, so their time is being squeezed from all directions. So developing tools not only to help the oncologist manage this dearth of information, but radiologists managing their time so they can focus on patients to improve the quality of experience of patients in a very anxious time is extremely important. So we're developing a number of platforms, cloud-based platforms and also on-premise platforms for the integration of data sets to make MDT performance and efficiency much improved in the coming years. And to give you an example of how things are moving, and I'm sure you'd be very familiar with these developments. In the past, as I said, the referral and diagnosis in oncology was very much a serial and binary approach. A patient came in to the oncologist, the referral took place, a scan was ordered, and the patient was then referred back once the data had been obtained on the image. The challenge now can be illustrated here. We have radiological imaging, all of the modalities that I mentioned, pathology coming in from biopsy, from blood tests, of course, cell analysis, also serum chemistry, all coming in. It's needing to be assimilated by the physicians. Omics data now, increasingly we see genetic matching of patients to treatments. And then of course there's the demographic, the electronic medical records. All of these factors can play a part in the diagnosis. What we're doing at GE is, as I said, leveraging machine learning and big data to integrate these data sets through cloud and on-premise systems to determine the right treatment for the right patient. And it's an integration of all of these pieces in the MDT team that we believe is going to facilitate that speed of diagnosis and of course the speed to treatment as well. We have examples already of predictive analytics that can leverage these features from pathology, from imaging, from demographics. We have examples of algorithms that can predict diseases like Alzheimer's in the same way and with the same diagnostic performance as imaging agents. We have algorithms that can predict whether patients will require stenting or revascularization following CT angiography. In the case of the work we're doing with Vanderbilt, we have algorithms that can predict which patients are going to be susceptible to hospitalization or who are going to require hospitalization in response to immunotherapy. So having the ability in advance of seeing which patients with confidence are at risk and then monitoring those patients more carefully could be giving an extreme advantage to physicians. So the portfolio that we have on the imaging side is actually focused on breast, prostate, colorectal, lung, actually colorectal and liver, as well as lung. And throughout the process and care pathway for all of these cancers, the platforms that we have developed, Edison, which is a GE platform for on-premise use in hospitals, and Navify, which is a partnership platform that we have developed with Roche, integrates these multimodal data sets, not only to support communication between physicians and caregivers in the multidisciplinary team setting, but also can act as a platform for the aggregation, integration, and subsequent analytics to deliver a more precise and informed process of diagnosis and treatment selection in the future. So this ability to provide a platform for advanced clinical decision support is going to be realized through these technologies. So to conclude, I started with saying, look, we're trying to develop solutions for our customers, solutions that can inform on the speed of diagnosis, speeding up treatment, and with a view to improving outcomes. This is essentially the heart of precision health. And what I believe is going to happen in the next few years is the leveraging of all of this technology, these advances in new products, selection of patients for the right kind of treatment, stratification, prognosis of patients with respect to their side effects, prognosis with respect to their treatment outcome, but deployed in a way practically that can make a difference on the ground in hospitals and in clinics. And then wrapped around that whole ecosystem is the ability to collect all of that data into a single system for easy reference, easy communication, and direct impact on improved patient outcomes. So it's our aim at GE to really help you and help patients improve clinical efficacy at every step of this pathway so we can deliver improved patient outcomes through precise and individualized therapy to improve outcomes. And I'm very grateful for the opportunity again to my colleagues here for allowing me to go first. I had a wonderful week, and just thanks to everyone involved in this, and I hope this presentation has been informative to what we're doing. So again, thank you for your attention.
So the question is about Gustave Roussy Center in Paris. So before you started this initiative regarding the reducing time per patient to 1 day, they had a certain amount of units of equipment, they had a certain patient pathway, whatever. So did it take a lot of work to establish such a workflow? I mean, sorry for interrupting you. Yeah. I mean, for an average hospital, for example, in Russia, would it take a lot of work to build such a process?
No, I don't think so. Gustave Roussy were fortunate to be able to build a new building. They had a building and they were able to construct an ideal workflow for the patients coming in, getting registered, and then being supported with a nurse navigator through the entire diagnostic process. They had that ability. But frankly speaking, it's not so much the organization of the rooms and the logistics, it's the processes and the procedure that is important. And what we have in collaboration now is access to those standard operating procedures, the scheduling, the kind of training that needs to be put in place to support that process, and of course the technologies that go in to make the diagnosis at 99% sensitivity and specificity. So actually it's more of a process re-engineering than a complete new clinic. Of course, where there is a possibility of a new clinic, it's an advantage, but actually the re-engineering of the process and advice and to support and hold hands basically to provide that information is really all that is required.
Thanks a lot.
Еще будут вопросы? I have one question. Could you tell us more about GE Healthcare new items or new technologies in the field of labeled antibodies production, I mean something that we could produce now using cassette fast lab system.
Yeah, so on the Fast Lab, I presented cassettes that have been developed for specific production purposes. We also have many recipes for blank cassettes, so cassettes that can be configured for bespoke or new radiochemistries. So this so-called developer cassette allows you to take recipes, radiochemistry recipes for example, labeling of antibodies or mini-bodies with chelates for zirconium, for example. So we have the ability to support that process. And in fact, we're working with several pharma companies who are interested in understanding the pharmacokinetics of antibodies or mini-bodies or affibodies, and understanding the pharmacodynamic uptake and dosimetry of antibodies. So we have those kinds of protocols that we can support labeling to be done. And this is extremely important in the selection of new antibodies like immunotherapies, selecting the right immunotherapy based on pharmacodynamics, pharmacokinetics, and of course biodistribution into the tissues is extremely important. And this is why I think your question is important. We can help with radiochemistry and nuclear medicine. We can support that process very effectively, I think.